Good evening to everyone who is joining us from Australia, and perhaps good afternoon and good morning to some of you who might be joining us from across the world. So thank you. I welcome to our Science Monash Alumni webinars. Today's webinar is being presented by the School of Science in Malaysia. So we've got some presenters, and also we've got the head of school who will give us a little bit of an update about the school and where it's located, and also what's happening over there. So before I get into the session, let me acknowledge the traditional owners of our land and pay respects to our elders, past, present, and emerging. And if there are any members in the audience who are Indigenous or Torres Strait Island members, I welcome you all to the country. In terms of today's session, we're going to have a bit of a school update from our School of Malaysia, Science in Malaysia, and we'll then move on to our presentation, which is about 30 minutes, and then it'll be followed by a Q&A session, which will give plenty of time, about 20 to 15 minutes for questions from the audience. I'll request our audience members to put in your questions as they come to you in our Q&A section. And when we do come to the Q&A session, Joshua, who's joining us today, who's a PhD student of Professor Qasim, who's today's presenter, he will actually moderate the session. So let me start off the session by welcoming Professor Emily Goh. So she is our head of school from School of Science in Malaysia. She's an expert in electroanalytical chemistry and has been, teaching and doing various research, and now being the head of school for about 17 years with the School of Malaysia. So she has very endearing connections with our alumni, and I'm sure lots of them who are joining us here will have the same sentiment towards you. So welcome, Emily. I'll let you give a bit of an overview of the school.
Thanks very much, Udi, for the kind introduction. Indeed, I'm very pleased to see some familiar names who are alumni, and of course, it's our pleasure to stay in touch again at this session. Before I move on to the update, let me just quickly share my screen, and if everyone can see the screen well. It starts off with School of Science. Is that okay, Udi? All good? Great, thank you. So if you may allow me to have the first couple of minutes to update you about the School of Science, perhaps it's also introduction to some of you who may be new to the School of Science at Monash, Malaysia. Before I move to that, I would like to bring you through the slides, and I would like to introduce you to our campuses. These are our international campuses. As many of you know, we have several campuses in Australia, we have in China, and I am here in Malaysia, Indonesia, India, and in Italy. With regards to Monash, Malaysia, we also have campuses here in Kuala Lumpur, and that's where the main campus is. And we have the School of Medicine in Johor Bahru, one of the state in Malaysia, and Segamat. So about our campus in Malaysia, we have established since 1998. And throughout the journey, we have gained recognition from the Ministry of Higher Education of Malaysia, of gaining some of this recognition. As you can see here, a self-accreditation status. MIRA is our research excellence status, and we are here today in 2023 celebrating our 25th anniversary at Malaysia. So Monash University Malaysia is Monash's third largest campus and one of Malaysia's most respected university. We are here as a platform for scholarly engagement with the Southeast Asia and beyond. So our Monash community, at the moment, we have approximately more than 9,300 students from various countries. We have more than 860 staff from the various countries as well. We have about 84% of our academic staff who holds the doctoral degree. So this is how the size of Monash Malaysia. And moving on to the next slides, that I will introduce to you our humble School of Science over here. We have 38 academic staff, and we are 100% of PhD holders here, 28 professional staff, and the school has about 700 plus, I think currently about 760 undergraduate students, and also about more than near 100 now postgraduate students, and a student-staff ratio is 20:1. And our student diversity, just to let some of you know that we do have students from different countries. I think this is how rich our culture is, even though, you know, we are located in Malaysia. I think this is great. About the School of Science and what we do, what we offer, very quick update. We have 4 courses now, mainly the Bachelor of Food Science and Technology, being accredited by the International Union of Food Science and Technology. Bachelor of Science, we have several majors, about 6 majors over here on the right, as you can see. Bachelor of Medical Bioscience, and the latest to the school is the Bachelor of Applied Data Science, which will be offered in July 2023. And we also offer pathways to research. So the undergraduate course 3 years, 4th year will be a Bachelor of Science Honours, and students can choose to pursue Master of Science or Doctor of Philosophy at our campus. And we do have several research teams over here. Would like to elaborate it further in my next slides, that these are the research area of our academic staff currently we are embarking on. Chemistry, food and plant sciences, health sciences, tropical and environmental biology, genome and genomics, and bioinformatics and biostatistics. And I believe you will hear more updates from Prof Qasim later on in this area of genomics. So there is a quick QR code if you are interested, but you know, in the interest of time, you may also visit our website for more information, or keep in touch with Udi if you wish to know more about the School of Science. There's a very quick one on to update that our research facilities has increased tremendously throughout this year. This few 10 years, I would say, in the past 10 years, we have several facilities in applied chemistry, in life sciences. These are some of the equipments just to keep everyone updated. We also have some equipment in the food science area as well. And one of the world-class infrastructure which we facilitate is the genomics facility. And Prof Qasim, of course, is the director of genomics facility in the School of Science. So the genomics facilities is ISO 9001 certified, and it provides our researchers with moderate to high throughput nucleic acid sequencing services. It hosts equipment, latest equipment such as PacBioSequel 2E and several other equipment, and I believe you will hear more from Prof Qasim himself. So that's all I would like to keep everyone informed about the School of Science at the Monash Malaysia campus. Do visit us on our website, or we also do have a Facebook. Just search us on School of Science, Monash University Malaysia. Thank you very much for the opportunity to give you a few words. I hope you will have a fruitful discussion with Prof Qasim, and I wish everyone a good evening. Thank you.
Thank you, Professor Emily. That was quite, hopefully for our alumni, quite an informative sort of introduction to the School of Science in Malaysia for people who might not have known that we've had a campus of Malaysia too, and the facilities we've got. So let's not take further time. We'll get into the session. Let me introduce you to Joshua Law. So Joshua was a PhD student of was an honor student of Professor Qasim, and now he's doing his PhD work here in Australia. So his works in cancer genomics at the Monash Health Translational Research Facility, and he's doing a lot of work in the breast cancer area, looking at history of breast cancer, looking at heritable risk factors, especially for women with a strong family history of breast cancer, using different types of data, such as whole genome sequencing and methylation data, etc. So they're all in the genome sequencing space. So Josh, I'll let you take over and moderate the session with Professor Qasim.
Yep, thanks, Uli. Good evening, everyone. It's my pleasure to introduce to everyone Prof Qasim. Prof Qasim is the Deputy Head of School in Research, and he's also the Director of the Genomics Facility in Monash University Malaysia. Prior to joining Monash University, he was employed at the world-renowned Wellcome Sanger Institute, and several of the Y chromosomal genetic markers that he identified through his research are routinely used in today's human forensic DNA analysis. His current area of research is in human genomic variations in health and disease, with a particular focus on genomics of indigenous populations from South Asia. He's also currently leading the Malaysian Biogenome Project with the ambitious aim of capturing the biodiversity of all complex life forms in the country through sequencing and promoting genomics for species conservation. Now, as his previous honor student, I've personally learned so much from Prof Qasim, and I could say that he has a great eye on detail, and I've learned so much from the aspect of how I should present my data figures, concise scientific writing, where to look for publicly available data, and how to utilize them in my research. He's also the kind of professor I aspire to be. He has a broad knowledge on various topics in human genomics, and is always capable of citing a paper seemingly out of nowhere, and that paper somehow is actually able to help guide me in my research direction. Personally, I like to say that Prof is, like, to me, the human Wikipedia of genomics. With that, I'll hand over the floor to Prof Qasim to share with us on today's topic, how genome sequencing facilitates our understanding of human evolution.
Prof Qasim, I think we'll get you still on mute, so I'll get you to come off the come online.
Can you hear me now?
I can hear you now. Yes.
Okay. And can you see my screen?
Yes, I can see a screen too.
Okay, so that's good. Just let me get a bit organized on my side. So thank you, Joshua, for that very kind introduction. As so kind of you and Emily to introduce me, and thank you, Udi, for organizing this seminar. So today I'm going to be talking about how genome sequencing facilitates our understanding of human evolution. It's a project which involves a lot of people across different continents, and also Joshua played a major role in some of the results that I'll be showing today. So the premise of my talk is that nothing in biology makes sense except in the light of evolution. This was a very influential paper that was written by Ted Dobzhansky, who was an influential Russian-American scientist. And he published this paper in American Biology Teacher in 1973, and really was made sense of the molecular synthesis of evolution and how we understand it. So briefly, I'm going to discuss basic genomics for those of you who are not from the science background. And then I'll dig into how we identify variations underlying genomic selection, and then talk about one interesting variation that we observed in East Asian populations, and talk about future directions of my research project. So basically, DNA is the genomic material. It's present in all cells of our body. It's composed of a chemical which has a phospholipase backbone, and a spiral molecule which is composed of four bases, known as adenine, thymine, cytosine, and guanosine. And we'll denote these by 4 alphabets of the English language, A, G, C, T, and that's the alphabets of genomics. So in in DNA, A always pairs with T and C always pairs with G. So if you know the sequence of one strand of DNA, you can guess what the sequence of the other strand would be, because A is always complementary to T and C is always pairs with G. So what we do in our lab is sequence the genome, and what you get is essentially a string of ACTGs in the sequence. And we spend the rest of our time trying to analyze what these sequences mean and what kind of function they code for. So remember, the genome codes for all the proteins, all the enzymes that are found in our body that make up the structures in our body and allow us cells to function in our body. And the 2% of the genome codes for these proteins. The rest of the genome essentially codes where these proteins are expressed and at what stage of development and life. So DNA is found in a specialized structure known as the nucleus, shown here in this green blob in a human cell. And the vast majority of the genome is present inside the nucleus. There's a small amount of DNA present outside the nucleus in the mitochondria, which is the energy powerhouse of the cell. And they're depicted here as these yellow dots. So the genome of the mitochondria is inherited from mothers to their children. So I will have my mother's mitochondrial DNA and she would have inherited it from her mother. So mitochondrial DNA delineates the maternal ancestry, whereas the nuclear DNA is a mixture of ancestry from both our biological parents. If you stretch the DNA inside our cell, it's about 1.8 meters long. And the way it fits inside the cell is it's wrapped around specialized proteins known as histones. And then these are formed structures known as chromosomes. So in humans, there are 22 pairs of autosomes or chromosomes that one set of which we inherit from our mother and the other set from our father. And there's one pair of specialized chromosomes known as the sex chromosomes because they determine the sex of the individual. So females will have 2 X chromosomes, males will have 1 X and 1 Y chromosome. The Y chromosome is inherited from the father, and so it will trace our paternal lineages. And the X chromosome is inherited from our mother. Females have 2 X chromosomes, one they inherit from the mother and the other from the father. The variation that I'll be especially talking about today is actually located on the X chromosome. So males would have 1 copy of that variation and females would have 2 copies. So sequencing is basically nowadays done by a process known as massively parallel sequencing, in which we sequence multiple molecules in parallel. So it decreases the cost and you get a large amount of data. And it determines the order of the bases in the DNA molecules that make up the chromosomes that I just showed you. The sequencing technologies currently are divided into 2 main groups, short-read technologies and long-read technologies. So these are analogous to, if you imagine that the sequence is can be assembled like a jigsaw puzzle. So short-read technologies are smaller pieces of the jigsaw puzzle and long-read technologies are larger pieces of the jigsaw puzzle. So with the long read, it's easier to assemble the genome or the puzzle, whereas with short read technologies, it's a bit more trickier. The market leader is currently Illumina, which is an American company that produces short read sequences. But at Monash, Malaysia, the genomics facility that Emily just mentioned, we have both the long read technologies, the PacBio and the Oxford Nanopore. The Oxford Nanopore is essentially a portable long-read sequencer. You can take it out in the field, and it's also been taken to the International Space Station where people have done some sequencing. And the other one is the Illumina MySeq that we have, which is capable of short reads about 300 base pairs. Regardless of the technology we use, the steps for sequencing are the same. You extract and fragment the DNA from the cells, particularly for the short read sequencing. Then you prepare a library of your DNA fragments that have been obtained from the cellular DNA. And then you essentially sequence those fragments, and that can range from anywhere from 36 base pairs to about 1 to 2 million base pairs of contiguous sequence based on the long read sequences. Once you've gotten the sequence, then what you do is you try and assemble the fragment if you have a good reference to map to, such as the human genome has a very good reference that took 20 years to establish, what we do is we just map those reads to this reference genome. Or you can also, if you've got long reads, do a de novo assembly or assemble the sequences from scratch by looking at where these fragments overlap. Once you've established the assemblies from different individuals, you compare them with the reference or with each other and call the variations as DNA variations. The DNA variations comes in all kinds of shapes and sizes. They can be very large scale, including like whole chromosome duplications. The best example of that would be Down syndrome, in which you have 3 copies of chromosome 21. At the other scale, you've got very small scale mutations, such as a single base pair substitution, such as the C changing to a G over here. And these are much more common in the genome. If you take 2 random human individuals, they'll be about 99.9% similar. And there will be around 3 to 4 million bases that are different between 2 individuals. These single nucleotide polymorphisms are also classified as SNPs. And these are the ones that we focus on when we analyze population genomics. And so in population genomics, we look at these mutations. For example, in this 22 base pair sequence I'm showing here, the C over here is changed to an A, shown in green. And so these are known as single nucleotide polymorphisms. They have characteristically 2 alleles. So alleles refers to one of the possible variations of a given locus at a particular position in the genome. So in this example I'm giving over here, this SNP will be located in a particular position in the genome. It'll have a C allele and it'll have a allele. So it is biallelic. SNPs are very useful for population genomic analysis because they have 2 alleles. And we assume that seldom the same position is mutated, although as we sequence millions and millions of genomes, we are finding those. The other feature is that the mutation rate for these variations is quite low. So you can actually conduct population genomics kind of analysis. And you can also deduce the ancestral state from outgroups. I'll just explain that in a bit more detail. So the variant allele in a common ancestor would be referred to as our ancestral allele. And the state after the mutation arises is known as derived. So for example, this is a mutation denoted by the reference number 1343879, which basically refers to a particular position in the genome, which in this case is on the X chromosome. And the mutation that we find in humans is a C to A mutation, which changes the protein sequence. So when we align this sequence that we obtained in the human samples with all the other primate sequences shown here, such as the chimpanzee, gorilla, and orangutan, you can see that the C position, shown over here in yellow, is pretty constant across all these primates. So this gives us very high confidence in saying that the common ancestor to the human lineage would have had C at that position, and A is probably the derived mutation found in the human species. So with that in mind, we basically sequenced genomes from the 1,000 Genomes project. So this was a project that was initiated by the Broad and the Wellcome Trust Sanger Institute, where I was working. And they sampled 26 worldwide populations. They are shown color-coded by the continent of origin of these populations. So the East Asian populations that I'll be focusing on are shown in shades of orange, which refers to the Chinese Dai population, the Han Chinese from Beijing and southern China, the Vietnamese population KHV, and the Japanese populations JPT. And so although it's known as the 1,000 Genomes project, we actually ended up sequencing 2,000 individuals. And the reason for that was because the sequencing costs decreased as the project progressed. And roughly, we identified about 85 million SNPs from these populations. So when we look at population relationships based on these SNPs, and this is a plot that was developed by Fadila, who was a master's student under my supervision. And so what we show on top over here is a scatter plot. Each dot over here represents an individual from a particular continent that's color-coded according to shades of yellow depict Africa, shades of green are European, shades of blue are South Asians, shade of orange and yellow, orange over here are East Asians, and the American populations are sort of scattered in pink across this scatter plot. So on the x-axis is the principal component 1, which is basically a statistical technique used to reduce multi-dimensional data to small components. And these smaller components explain part of the variation that we observe in the data. So the first component on the x-axis, which explains about 51% of the variation in the data, separates African populations in yellow from non-African populations. And then the y-axis, which is the second dimension, accounts for about 25% variation in the data, and it separates all the non-African groups. So this is consistent with genetic and fossil and archaeological evidence showing that human populations, modern human populations, or humans that look like us and have a skeletal morphology similar to us, originated in Africa. And the African populations are more diverse because they are separated across the first component over here. Also, as you increase geographic distance from Africa, you see there's a decrease in genetic diversity, such that European, East Asian populations have a lesser genetic diversity than Africans. As you go out of Africa, you see really divergent populations such as Europeans in green and East Asians in orange. All other human populations fall on a line between these populations. For example, the Singapore populations, or the Malay populations are closer to East Asians, or the orange dots, whereas South Asians are more closer to the European populations, or the green dots over here. The Singapore Indian populations falls right on top of the South Asian populations from India, Pakistan, Bhutan, and Nepal, and which is not surprising because these populations were recently settled in this Southeast Asia. And so the Malay populations have a substantial East Asian admixture. That's why they are closer to populations from East Asia. The reason American populations are sort of scattered in the plot is because they have substantial European admixture and African admixture because of the slave trade and the colonization by Europeans. At the bottom over here, I'm showing basically an admixture plot, which is in which the same individuals are represented by vertical lines, and the color-coded basically represent genetic components that are shared. So you can see that continental populations share a lot of the similar genetic components. So based on genetic data, we can surmise that human populations basically arose in Africa and then spread rapidly across outside Africa. The time of this spread and the migration and the route that they took outside Africa is still quite debated. It's not very clear right now. Just recently, actually, just this week, there was a paper that showed that modern human bone was discovered in a cave in Laos, and it was dated to around 70 to 80,000 years ago. So basically, we think that humans left Africa and very quickly traveled all across South Asia, Southeast Asia. They were in Australasia around 55,000 years ago in central Australia. But archaeological evidence suggests that they reached the Australian subcontinent around 65,000 years ago. The last continent to be inhabited by humans was America, and a population of Northeast Asians migrated across the Bering Strait into North America and then quickly spread into South America. And that was the last continent that was populated by modern humans. There was a separate East Asian migration which bypassed North and South America and went straight into Greenland. And these were the humans that are closer to current day populations of Eskimos and Inuits living in Arctic, in subarctic geographical areas. Also, humans, when they moved out of Africa, they encountered other archaic human samples or human beings. And these archaic populations have been discovered both by genomic sequencing, and for Neanderthals, there's a substantial evidence, fossil evidence, particularly in Europe. We think that humans, once they left out Africa, did mate with these Neanderthals somewhere, most likely in the Middle East, because all non-African populations of modern humans have about 2% Neanderthal admixture. Intriguingly, there's also another archaic populations identified from genomic sequencing of samples that were obtained from Denisovan Cave, which is a cave in Siberia in Russia. There is actually a higher mixture of this genomic phenotype in populations from, particularly from Papua New Guinea and Oceania, which leads us to speculate that this admixture most likely occurred somewhere in Southeast Asia. And as humans settled in these areas, they also adapted to different climates, diets, and pathogens. And so these adaptations left signatures in our genome. So how do we actually detect these signals of positive or Darwinian selection? So let's suppose that a mutation arises in a human population, let's say that gives us blue-colored skin, and then it's beneficial in some environments. So these individuals who have this blue-colored skin will increase in frequency, and the process by which this advantageous mutation spreads in a population is known as positive selection. And as Darwin surmised, even though he did not know much, nothing was known about genetics at the time he proposed this origin theory through natural selection, he stated in that it's not the strongest of the species that survive, nor the most intelligent, but the one that are most responsive to change. And essentially what we think in modern synthesis, it's the individuals that have variations that can respond to changes in climate, diet, and pathogen exposures. So how do we detect these signals? So let's suppose there are two populations of, let's say, a population of East Asians on the left side of the slide and a population of Africans on the right. And they're separated by a genetic barrier, such as the oceans. The blue bar over here represents the genomics segment. So this would basically be a sequence of DNA. And the circles over here represent SNPs, or single nucleotide polymorphism, or substitutions. So let's suppose in East Asians, there was a star SNP, which represents a beneficial SNP. So what will happen is that this SNP will increase gradually in frequency in the population. And this increase in frequency of the population leaves certain signatures, so that when we examine the population in the present, what we'll observe is that the East Asians have this star SNP, which is at high frequency. It's not present in the African population. You'll have other SNPs which are in close vicinity to this star SNP, such as the purple one and the orange one over here, which will increase in frequency in East Asians as well, known as genetic hitchhiking. You don't observe that, so just random allele frequency fluctuations will occur in Africans. So the orange one will be at low frequency. In addition, Africans will have other SNPs which have risen in frequency, which will be present in low frequency in the East Asians. So these signatures can be detected by population differentiation. So like differences in derived allele frequency or fixation alleles. You can also look at extended areas of linkage disequilibrium. Essentially, that means is SNPs that are nearby will also increase in frequency. And this linkage will be broken down only by recombination that occurs in the sperm and ova cells before the next generation evolves. And so it takes a long time for this linkage signal to be broken down. And that can be detected using certain tests. And then also the site frequency spectrum gets disturbed, so that when you compare the site frequency spectrum between a selected region and neutral region, you would see specific signals. So the most extreme of these signals leave the strongest signatures, and we can use these methods to detect all these signatures. So using these methods, we actually developed a tool to prioritize variations that we could then study. And basically, the tool that we developed looks at derived alleles that are common in a population, so more than 10% frequency. They are very specific to particular populations and have a strong predicted functional effect. So FindMap utilizes variant annotations that were actually designed to predict how damaging a mutation is or how pathogenic it is. And so it was focused for looking at disease-causing mutations. We are actually using it to identify mutations under selection. And so the FindMap will look at alleles which are high frequency, derived in specific populations, and pathogenic or deleterious in some way. The tool will not identify all positively selected regions in the genome, but will at least look at the extreme outliers that we can focus on for further detailed analysis. So the hypothesis behind this is that alleles which are predicted to be deleterious should have been removed by negative selection, so they should not be able to reach very high frequency in populations. But if they are predicted to be harmful and they are high frequency in a population, it's possible that they're not harmful and they probably could have some adaptive value in our evolutionary past. And so looking at the East Asian populations that I showed you just now, we identified several mutations and we were really happy to see this graph. So this graph shows the chromosomes on the x-axis, and on the y-axis is the FindMap score. So the higher the score is, the more pathogenic the variant is predicted to be, or in our case, the more adaptive the variant is predicted to be. And so we saw EDAR, which is known signal in East Asians. We saw ABCC11, which is a known signal in East Asians for earwax. And so we were intrigued by this MAGI2, which has been known for a while, but nobody knew what the function of this gene is. So Joshua, when he was a honors student, explored this in a bit more detail. He identified that this gene codes for a protein which is known as a melanoma antigen gene. And it's known as that because it was identified in a human melanoma cell line. It's present on the human X chromosome, but there was no known function. The gene codes for a protein that's about 523 amino acids long. It has these two melanoma antigen domains shown in green and blue over here. The mutation that we looked at gets rid of around 400 amino acids. And so it truncates the protein around 120 amino acids. And so we think it's a very damaging mutation. We also know from the population genomic data sets that Joshua looked at, the allele frequency for the loss of function MAGI2 allele is very high in East Asian populations. Actually, over 90% of the Japanese population have the allele which gets rid of, more or less, knocks out the gene. When did this first appear? We looked at ancient DNA data, and Joshua saw that this allele appeared in Eurasia around 40,000 years ago, but then rapidly increased in frequency in the past 10,000 years, particularly around 7,000 years ago. So that this is a graph showing the selection signal. So if you can see the steep line above this dotted red line, that is when the signal increased. And this occurred around 7,000 to 8,000 years ago before present. And so this most likely occurred during the Neolithic period in East Asia. What does the gene do? So we know from some studies in human cadavers that the gene is expressed in human brain tissue. So what we did was, with the Wellcome Sanger Institute, we generated a knockout mouse for this gene. So the mouse did not have a functional MAGI2 gene. And we then got collaborators in France who looked at the brain tissue histological slides. And they actually showed over here that in male samples, there was particular areas of the brain that were larger in size as opposed to the wild type mice. And so it told us that there was most likely this gene had a function in brain tissue. When we examined the mice itself, physically, there was no difference between the males and females, and the mouse without the gene behaved perfectly normal. So we could not detect any discernible phenotype in the mouse. So then we got collaborators in China who looked at MRI scans of human individuals who were genotyped for this SNP. And to our surprise, what we noticed was that this inactivated or individuals who had 1 or 2 copies of the inactivated MAGI2 gene, in males, it resulted in a larger brain size, both the gray matter and the white matter, whereas in females, it actually reduced the size of the white and the gray matter. And this is something that was puzzling, and we really were not expecting such drastic differences, although it replicates exactly what we found in the mouse histological pattern as well. And this was very highly significant. For the white matter, we observed the same thing. This was just in a sample of around 140 individuals. So it's not a very large sample size. And then what Joshua did was we had a sample of around 72 Malaysian individuals in whom we'd done some cognitive testing. So basically, we did 3 tests. We had asked individuals to memorize a list of 16 words and then asked them to recall those words. Then we asked them to do basically a task in which they assembled 3 colored balls on 3 different pegs. And we told them to repeat this about 16 times. So it basically was a measure of their higher level cognitive functions. And then after they completed the task, we asked them to recall the same 16 words that we'd given to them earlier. So these tests were recorded, the time it was taken for the individuals to recall these words, and also the time to complete the Tower of London task was measured. And then we essentially looked at the genotypes of these individuals, and Joshua tested if there was any significant difference in individuals who had the loss of function allele or did not have the loss of function allele. And what we were able to observe that the people who had the loss of function allele had a higher short-term word recall score than those who did not. And we don't know how this would be of evolutionary advantage, but we do have a few theories. So one of our theories is that it was something to do with the transition from a hunter-gatherer lifestyle into an agricultural or Neolithic lifestyle that began about 10,000 years ago in East Asia. Our other theory is that it has something to do with the origin and expansion of the Chinese language around 7,000 years ago. Both of these have pros and cons. So for the transition, we know it fits in very well with the sexual dimorphism phenotype that we observe in hunter-gatherers. But the transition to sedentary lifestyle was not East Asian specific. And so why was this gene selected only in East Asians and not in other populations? So for that, we try and explain it through language, because Chinese and Sino-Tibetan languages overall are logographic and they require good spatial memory. We also know that there is sexual dimorphism in the way males and females process language. And it could explain this East Asian specific expansion of this nonsense SNP. But I mean, language is spoken both by males and females. So why would there be sexual dimorphism is difficult to explain. And also, it's also difficult to disentangle linguistic affiliation and population genetic background, because MAGI2 is not the only SNP with this pattern. There are a number of other SNPs that could equally explain this pattern. So now what we're looking at is studying the association of this SNP in a very large data set. So we're in the process of collecting these large sample sizes, which would be important to do these association analysis. Other groups in China are also collaborating to investigate the 3D structure and spatio-temporal expression of this gene in adult and embryonic samples, and also in vitro and in vivo models. And basically, we're trying to unravel what this gene does, how it responds, and how it responds to changes in the genotype or the loss of function allele, and what is actually the function. So in conclusion, I've shown that humans have adapted to different environments as they moved out of Africa, and particularly we observed this population-specific variation in MAGI2, and the loss of function allele is associated with differences in male and females in both mouse and human brain sizes, and it's found at high frequency in populations across Asia. We are conducting further population studies to understand its role in human cognition. So if you want to read more about these, there are some papers that have already been published, and we're in the process of submitting Joshua's work for publication. I think all this work would not have been possible without collaboration across different continents for teams in Malaysia, a lot of my students involved in it, also teams in France, Sanger Institute, and China, and also we thank the NUS for sharing the genome sequencing from the Singapore dataset. So that's all that I wanted to talk about today, and I'll hand it over back to Joshua so that he can moderate the question and answer session. Thank you all for listening.
Yeah, thank you, Prof Qasim, for the wonderful presentation. Yeah, and it's good that you've covered basically all bases from the basics of DNA to the sequencing technologies that read the DNA to evolutionary theory and how it all ties to the MAGI2 project.
Mm-hmm.
And yeah, it's a really interesting presentation. With that, I'll open the floor for any questions. Okay, so there is already one question from an anonymous attendee.
Mm-hmm.
Are there any evidence for Caucasian race? If so, who are they and how did they migrate and spread?
So I mean, they're the only, what we call Caucasians, present day populations, which originated in the Caucasus Mountain, is really an admixture of individuals. And our best examples really come from European ancient DNA. And what those data have shown is that present day European population is a mixture of original hunter-gatherers who inhabited the continent after we moved out of Africa. They got admixed with farmers, most likely coming in from Anatolia or the present day Turkey. And then this population was then admixed with another group coming in from what is present day Siberia or regions between Caspian and the Black Sea. They had a particular culture known as the Yamnaya culture. And so present day European populations are basically admixtures of these three groups. We see similar admixture in populations from South Asia, in which they have an ancestral North Indian and an ancestral South Indian group, which is essentially hunter-gatherers from the first African migrations mixing with farmers, in this case from Iran, and then individuals from the Yamnaya culture. So, I mean, modern human populations are very admixed. This was even before there was travel by air. And so we've been mixing together with other people. And I think there's no such term as a race. It's a very fluid term right now based on genomics. I hope that answers your question.
I hope so too. Okay. The next question from Melissa.
Mm-hmm.
Hi, Prof. If funding and logistics was not an issue, which population would you most be interested in studying, and what would your study focus on?
So I guess what is interesting being in Malaysia is there's a lot of populations, particularly the indigenous populations known as the Orang Asli, about which we know very little. And we've only sequenced maybe a handful of these individuals. And so it would be really interesting to sequence a lot more of these individuals. But what we'd also want to do is at the same time do a lot of phenotypes measurements. So measure maybe the heart rate, their basal metabolic index, look at their height, weight. And so we can actually then use these populations as model organisms and try and do genomic association studies in large population data sets. So I think Southeast Asia as a whole is a really interesting region to explore these populations, particularly the isolated populations. I mean, the other interesting population for me would be the Hazara population in Pakistan, which is another isolate population with substantial East Asian admixture.
Okay. The next question from Tuan Le. Is the MAGI2 gene on the X chromosome? If so, would the effect of the MAGI2 mutation be more pronounced in hemizygous males since they only have one X chromosome?
Yes, that's exactly what we observe. So males who have just one X chromosome, if the gene is knocked out, they really don't have any MAGI2 protein. And that is why we see the differences in the male and female who have the loss of function allele. Because the females, if you have one gene knocked out, you still have one functional copy. But the difference in brain size that we observe is females who have both copies of the gene knocked out, they have a smaller brain gray matter and white matter, as opposed to the males in which just one copy of the gene is knocked out, but they have a bigger size of the gray and white matter. So that's a very paradoxical situation, which is really interesting to follow up on. So yeah, if the gene is on the X chromosome, then if it's knocked out, males will not have a protein product. That's exactly what will happen.
Yeah, and that is probably also the basis for the sexual dimorphism.
Yeah, that's true.
Okay, next question, Dr. Farhina. What would be the justification when an allele that is assigned as mutant in a database is found the most prevailing in a specific population in normal individuals?
So I think there are two reasons why allele frequencies increase or decrease in a population. One is just random fluctuation of these alleles. So let's say the mating population had a larger proportion of individuals with the mutant allele. And so if that happens, then just by chance, you will get increase in frequency. The other way you can get an increase of frequency of pathogenic variation is if the populations are isolated. So if they've undergone a bottleneck and the mutation is not lethal, so it doesn't kill you before reproduction, then from the evolutionary point of view, it doesn't really matter. It can increase or decrease in frequency. The other way is if there's selection that happens on a particular mutation, so that can increase the frequency of the mutation fairly rapidly. And that's what we observe in the case of MAGI2 loss of function mutation.
Yep, I think that answers the question. One more anonymous attendee. Hi, Prof. Thank you for an interesting talk. What is your approach to sample collection? Is the ethics process complicated?
I think getting the ethical approvals for such large studies is always complex. I think the 1,000 Genomes project had a really unique approach in which they actually, from the onstart, they involved lawyers, lay people from the medical background and people with non-medical background to get them involved in the sample collection. And then they also took into account individuals from the community itself where they were sampling. And so that, and they came up with an informed consent form that clearly explains that they were going to do whole genome sequencing and the data will be made publicly available. I think what we need to be concerned about is that if we're doing medical genomic sequencing for medical purposes, the risk is that with the whole genome sequence, even if the researchers don't want to, but anyone not in the research team, if they're really interested, they can, from whole genome sequences, dig down and identify individuals. And so I think that's the ethical risk and that we need participants to be aware of, and also ethics committee to be aware of. And so that does make getting samples a bit tricky. But I think as long as you're upfront about it and make sure that your participants understand the risks, because they're not only decoding their own genome, they're also decoding genomes for their close relatives and also their descendants, and will also find out information about their ancestors. So I think we need to make that very clear when we do collect samples for whole genome sequencing or genotyping. So it is difficult getting ethical approvals, but with these safeguards in place, I think we can take everyone on board and make sure they're aware of the risks and the benefits.
Yeah, I think ethics in human genomics is a very important thing to discuss on as well. Like even when I'm working right now in hospital, like clinical settings, I think a lot of it, like for example, from like how do we store whole genome sequencing data from patients and all that, it's really important. And also like the whole protocols have to be like listed out for the participants. So yeah.
Yes.
Okay, so the next question, okay, it will be our last question since we're running short on time. Anton, during your initial studies, have you found any pathologies associated with these MAGE2 mutations? What is the possible overall advantage of having this mutation apart from the cerebral size?
So Anton, the mouse we studied in great detail, and we were, except for this difference in brain sizes, we really were unable to identify any other pathology in the mouse. So, it could be that the selection was nothing to do with brain. It maybe was some other phenotype, which we haven't really studied in detail now. And maybe it's no longer important from the evolutionary point of view. And so the selection sort of relaxed. But at the moment, our best guess is it's something to do with brain function. But we can't be sure that it was not because of other reasons. But for the mouse model, we were unable to find any deleterious impact of this mutation in mice. And as far as we can tell, we don't see any deleterious phenotype in humans as well.
Okay, we still have time for one last question. Can we sequence DNA from the human fossil dated 80,000 years ago found in Laos? What is the oldest fossil from which DNA has been sequenced?
The oldest fossil is around 800,000 years ago. It's not human. It was if I remember correctly, it was I think it was from a horse. The oldest human DNA that's been sequenced around 50,000 years ago. We've been unable to extract DNA from any other samples that are older than that. And particularly, the samples which are well preserved in permafrost are the ones we get the best ancient DNA samples from. And the best samples we've gotten are from the skull bone, the petrous bone of the skull. And so if it's well preserved DNA in permafrost in Siberia, we can expect to get DNA out. In Africa, some people have managed to get some DNA from ancient samples as well. In Southeast Asia, it's tricky because of the environment. DNA is really easy to destroy in the presence of humidity and water, so it's unlikely to survive. But there have been some ancient DNA samples sequenced from Southeast Asia, and we hope that as more samples are discovered and we extract DNA from them, we are able to identify better the ancient DNA landscape in Southeast Asia as well.
Yep. Thanks, Prof, for the wonderful answers and also for giving a very good overview of human evolutionary genomics. So with that, I would now hand the floor back to Udi.
Thank you, Professor Qasim and Joshua, for moderating the session. That was certainly an interesting topic and lots of questions from the audience. There's still a little bit more questions there, but given the interest of the time, I think, we're going to wrap up the session. I thank all the alumni who's joined us for tonight's session, and we will continue to have more webinars from our researchers from the different schools of science in Monash Faculty of Science.